Hello, my name is Daniel Berenberg. I'm a PhD student in the Computer Science Department at New York University, where I'm also a member of the Beno Lab. Today I will be presenting our work on benchmarking feature extraction methods for structural comparison. So today I will first introduce the problem by defining structural comparison and describing our data. Then I will describe two contrasting methods for feature extraction. The first method uses manually crafted features, in this case so-called graphlets, to characterize protein structure. The second method uses an optimization-based machine learning approach to learn topological features. I'll then present a benchmarking study using the two methods that pertains to the feature's effectiveness in the context of fold recognition and structural representation learning. I'll then conclude with a few remarks on the two approaches. So structural comparison refers to the notion of assessing the topological similarity between protein structures. While two structures may not be identical, they may share common characteristics, in which case they should be deemed similar. Today, with the advent of fast structure prediction methods such as AlphaFold 2, TR Rosetta, and more, it is becoming increasingly more relevant to develop methods that scale well with large data sets. This means developing methods that are capable of alignment-free topological similarity detection. In order to do so, several methods have been proposed that, rather than directly computing the similarity between a pair of structures, instead they choose to embed the structure into a low-dimensional space in which the distance between two structures reflects their similarity. This is often done by first pre-processing the structure by converting it to its C-alpha contact map at some threshold and extracting meaningful features from the resulting network. As I noted earlier, extracting such features can either be done via a manual or a learned process. But once we have our feature extraction protocol, we can use it for several different downstream applications, including fold recognition, fold discovery, if the method was unsupervised and does not require prior structural labels, and structural comparison, and even protein function prediction. In this work, we are using the CATH database, which is a large hierarchically organized collection of structural domains. There are 4 tiers to the CATH hierarchy, each pertaining to increasingly more specific secondary and tertiary structural characteristics. Those tiers stand for class, which coarsely sorts proteins into either mainly alpha, mainly beta, a mix of alpha and beta, or little secondary structure. The second tier is architecture, which describes the shape of the secondary structural units. The third is topology, which refers to the fold of the protein. And the fourth is the homologous superfamily, in which evolutionarily related proteins sharing the same topology are further grouped together. Now, I will be presenting a benchmarking study using sufficiently sequentially divergent structures from the CATH database. As I described earlier, generally the methods I'm going to present using the use the C-alpha contact map as their input data structure, and this work is also meant to present a sufficient baseline for structural comparison methods. So I will consider 2 general classes of feature extraction: manually crafted and learned feature extraction methods. The first class of methods is that of manually crafted features, namely graphlets. So graphlets were originally introduced by Natasha Pajuli in 2006 as a method to assess the structural role of a node in a protein-protein interaction network. Graphlets are non-isomorphic structures that can be seen as analogous to wavelets in signal processing. And a node's graphlet degree vector is constructed by counting for each graphlet how many non-redundant instances of that graphlet that node participates in. This idea makes for a fairly simple method for understanding the role of a node in the in a graph. Graphlets have also been extended to incorporate canonical ordering or orbits in order to further assess a node's structural relevance. And although there do exist fast algorithms, obtaining graphlet degree vectors is computationally intensive, as we have to consider all substructures, all nodes, and in some cases all orbits. Since we are summarizing protein structure, we use graphlets in order to generate a so-called global graphlet degree vector, which is done by computing node-level graphlet degree vectors and collapsing it into a histogram of structural motifs, which is seen below. In our benchmarking study, we considered two different graphlet representations. Those computed by ORCA, which does not consider orbits, seen on the top right, and the representations computed by graphene, which does consider orbits and whose graphlets are seen on the bottom right. The second method is a learned or optimization-based feature extraction method that uses graph convolutional networks. Graph convolutional networks, at least the version seen here, were proposed by Kipf and Welling in 2017 and work by updating node-level feature representations by aggregating inside local neighborhoods. This is done by using a normalized version of the adjacency matrix representation to propagate node-level information amongst neighbors, as seen in the expression depicted on the top right and the figure depicted on the bottom left. This idea is viewed as a message passing scheme in which node-level representations are informed by the representations of their neighbors. The idea of message passing implies that in a long cascade of GCN layers, GCN standing for graph convolutional network, nodes far away from each other on the graph are still capable of influencing one another's representations by message passing. In practice, we have found that these methods require powerful node-level features in order to maximize their performance. We use graph convolutional layers in order to minimize a graph denoising objective. This is done by sending initial node-level features, say the residue identities for each node in the contact map, through a cascade of GCN layers and asserting that the final output must be capable of reconstructing the original adjacency matrix when multiplied with by itself transposed. So here we are looking at some node-level features given by a graph autoencoder. We multiply them with their transpose to obtain an n by n adjacency matrix, and we assert that the resulting matrix should be close in terms of Frobenius norm to the true adjacency matrix. We evaluate these methods with two different metrics. The first, called triplet scoring, assesses the effectiveness of the induced similarity metric as a binary indicator. We construct positive and negative pairs of proteins, that is, those sharing and differing in their structural label, and calculate cosine similarities. We use these similarities as binary classifications and hope that more often than not, proteins sharing the same label are deemed more similar than those that differ in their label. We quantify this by calculating the area under the precision recall curve of the classifications. Secondly, we calculate the mutual information, which is used to assess the degree to which proteins assigned to the same cluster by a k-means classifier, the degree to which they share the same structural label. That is, with this measure, we quantify whether there is a coherence in our space with the existing label space. Given that explanation, here I am showing the results of the benchmarking study. As you can see, I trained one graph autoencoder on contact maps and compared it to three manual methods: ORCA, which uses the unordered graphlets; graphene, which uses ordered graphlets; and finally, simply counting the number of clicks up to size 7 inside the structure. The number attached to each label in the legend on the left indicates the angstrom threshold used to calculate the embedding vector. Here we can see that manually crafted features greatly outperform the optimization-based method in both metrics, but I'd like to highlight that the graphlet-based methods quickly become very slow as we increase the density of the contact map by increasing the distance threshold. So to conclude, we conducted a benchmarking study using two types of topological feature extraction methods: manually crafted features using motif counting and learned features by optimizing a graph neural network. We saw that there is a significant gap between the performance of manually crafted and learned features. This can be seen as a significant opportunity for improvement. In order to scale to big data and keep up with the rate at which modeled structures can be instantiated, it will become necessary to sharpen the performance of a graph neural network so as to cut down on comparison cost. I'd also like to offer some explanation as to why graph neural networks perform so poorly, at least in this task. One fact that we can see is that the features are not hierarchical. That is, although there was some semblance of clustering at the topology level, the networks were not capable of picking up higher order structure features that might indicate information at the class or architecture level. There is also a growing body of work showing that there are limitations on the expressive power of message passing neural networks. In the work shown on the bottom left, Chen et al. Show that message passing neural networks are not capable of counting substructures beyond size 3. While counting substructures may not be the only effective policy for structural comparison, it has been shown to be quite effective with the graphlet-based methods. So in this case, we can say that even if graph convolution was performing well, the learned features would not represent something akin to the graphlet degree vector. I would like to point out, however, that the graph autoencoder does have some merit. Here I'm showing a result in which the autoencoder successfully separates immunoglobulin and Tim barrel folds. And in addition, a CATH annotated immunoglobulin protein, seen here, that is misclassified by the model as a Tim barrel, seems to present in its 3D structure Tim barrel-like properties. This suggests that mature graph convolution-based method might be able to correct mistakes made by the automatic CATH pipeline. Finally, I'd like to conclude with an outlook on the future of structural comparison methods and graph neural networks. Although we can see that on the right, every year there seems to be a stronger interest in and contributions to the graph neural network subfield, very seldom have we seen methods benchmarked against classical methods such as graphlets, which have been shown to perform at the state of the art, at least on this problem. Secondly, the protein structural benchmarks used in the literature do not seem to capture the true complexity of protein structure. Seen below in the table, the proteins dataset, which is a common benchmark for graph neural network methods, has markedly less samples as well as less nodes and edges on average per sample than our benchmark, which is gleaned directly from the CATH database, which is a reputable resource. More difficult benchmarks are critical in order to build more effective and useful methods. So thank you for listening to this presentation of a simple benchmarking study of structural comparison methods. This work would not have been possible without Rich and Vlad, with whom I work closely and have guided me through the project thus far. I'd also like to thank the Flatiron Institute and NYU for providing the computational resources and funding in order to conduct this research. And without further ado, I am happy to take your questions. Thank you.